Welcome everyone to this bi-weekly NSF Expeditions seminar series. For all our expeditions colleagues, Simon doesn't need any introduction, but since we have a lot of other folks who have joined us today, I thought I will briefly introduce Simon. Simon Levin is the James McDonnell Distinguished University Professor in Ecology and Evolutionary Biology at Princeton University, and the Director of the Center for BioComplexity at the Princeton Environmental Institute. I must note that his institute, the BioComplexity Institute, the same name as our institute. And we also have
who was first? Who was first?
Simon, without a doubt. But Rita really was the one at NSF who started this, as I understand. She's going to join us. So this is it's very nice that we have three different groups here interested in the same topic.
Since Moshe just interrupted constructively, I want to say that the Princeton Environmental Institute has recently changed its name to the High Meadows Environmental Institute.
So we'll correct that, Simon. So Simon was at Cornell for 25 years before moving to Princeton, and his work examines the structure and functioning of the ecosystem, the dynamics of diseases, and coupling of ecological and socioeconomic systems. I've been really amazed with the breadth of the work he's done over all these years. He's, of course, very well known, has received many accolades. He's a fellow of American Academy of Arts and Sciences, the AAAS, the National Academy of Sciences, and the American Philosophical Society. He's also elected to the American Mathematical Society recently. Simon also has won a number of other awards, including the Kyoto Prize, which is really prestigious, the Heineken Prize in Environmental Sciences, and the Tyler Prize for Environmental Achievement. Finally, he's a National Medal of Science winner in 2016. I guess it was awarded in 2015, but was formally given to him in 2016. For us, it's been a pleasure to have Simon as our colleague, a mentor and a friend for a long time. And I think the talk today promises to be a very interesting talk that really takes you through the journey of his work over the last, I would say, 40 years, 50 years. He can tell us how long this span is. So thank you very much, Simon, for doing this. Really excited. Rita just joined as well. Rita, welcome. And we'll go ahead and start.
Thank you very much for your kind introduction, Madhav, and especially for your leadership in this expeditions program. The title of my talk is on COVID-19 and what it's driven in terms of alterations in the way we model epidemics. I want to thank the various funding sources that have funded my work over the years, but especially the expeditions in computing that Madhav leads and that so many of us are part of, and those are the partners on the bottom. It's been an incredible and productive effort. So as most of you will know, the mathematical theory of infectious diseases has a rich history going back to Sir Ronald Ross, the Nobel laureate, more than a century ago, but especially built on the models of Kermack and McKendrick, which remain foundational, where they split the population into susceptible and infected individuals and wrote equations describing the transformation between them. And I'll say a bit more about these for people who are not familiar with them. So based on this, and over the years, a rich mathematical literature has grown. Here are two books that I would recommend to anybody who wants to get into this, especially the book by Anderson and May, now several decades old. And on the more mathematical side, a book of Carlos Castillo Chavez and the late Fred Brouwer, who sadly passed away a few weeks ago. Yet despite a century or more of elegant theory, new diseases emerged, like the pandemic we're dealing with now, and old ones like tuberculosis reemerged. And I thank Madhav for this slide and this editorial by Harvey Feinberg and Mary Wilson that appeared a short time ago, calling out the importance of infectious diseases as a research area, infectious disease modeling, treating the problem as one in complex adaptive systems in which individuals make decisions or engage in interactions and emergent patterns come about, and the need to take a multidisciplinary approach to these problems. Now, in terms of what the theory has done, one of the greatest advances in the theory has been advising vaccination strategies. But nonetheless, we still are having problems with diseases like measles that we thought we had conquered, at least in the US and in the UK. Years after elimination, there's a reemergence, and largely because of pockets of resistance that have developed to getting vaccinated. In the US, this will show you some of the outbreaks that have occurred over roughly the last 10 years, and largely because in various places in the US, there's been resistance. I'll show you those pockets later. And everybody's aware that COVID-19 is with us, and this has led to what I think was an unpredictable political division in the country in terms of getting vaccinated and misinformation that is spreading. And when I say spreading to other shots, I mean there are people now beginning to speak up, including people in the US Senate, arguing we shouldn't mandate vaccines in schools, for example, not only for COVID, but for things like measles, mumps, rubella, things that have been with us for a long time in terms of policy and are now being challenged. It would be disastrous if it broke down. So let's go back to the classic theory, just a very short introduction. I know most of you will know this, but the Kermack and McKendrick theory roughly splits the population, as I said, into boxes of individuals who are susceptible and infectious. Usually adds a compartment called remove, which means people who have recovered, people who have been vaccinated, people who have died. And usually there is, in this formulation, something called the latent, which you can think of as the asymptomatic stage. But in most models in the literature, it is discarded. But we've learned from COVID that that's a mistake. We can't do that for COVID. So just focusing on the equation for the infectious class, this is the simplest formulation of it. It says that the rate of change of the number of infectious is proportional to the number of susceptibles and the number of infectives. And then there's a death term mu and a recovery term gamma. Now, there are variants on this, especially in modifying the transmission term, the so-called bilinear term. The simplest extension would be to say, well, it's not the number of infectives, because otherwise, for example, New York City would have rates of transmission 100 times or more, let's say more than Charlottesville. But it's the proportion that is, and the truth is probably somewhere in between. So N here is the total population size. But there are other forms, nonlinear forms, that can be considered. But let's stick with this for today because it's what the basic theory is based on. And basically you want to know, is the number of infectives going to increase? So let's focus on the equation at the top. And that means there's a threshold for outbreak or endemic based on whether the right-hand side of that equation is positive. And we can algebraically rearrange that to say that that means that beta dividing by I, that means that beta S over mu gamma must be greater than 1. That is the number of secondary infections that arise from a single infection. And it's usually termed capital R, and here I denoted capital R sub S, because it, in this case, depends on the number of susceptibles in the population. The term you hear most often referred to occurs when the whole population is susceptible, when it's a new pathogen or new variant of the pathogen arising in the population. And we call that R naught when capital S, when the number of susceptibles or the proportion of susceptibles, depending on how you're modeling this, is equal to the total population size. And instead of calling that Rn, we usually call that R naught. And Rs is sometimes referred to as R effective. So if you want to deal with an outbreak, if you want to begin to get it to drop down, then what you want is to look at that quantity Rs, and there are various ways you could get it down. One, which is not acceptable, would be to increase the death rate, but the other, which is acceptable, is to increase the recovery rate, gamma, or to decrease the terms in the numerator. That means either decrease the transmission rate for example, by mask wearing or quarantine, or to decrease the number of susceptibles, either individuals who have already been infected or individuals who have been vaccinated. So those are the various ways that we expect an outbreak eventually to get under control. And that's why you hear all this discussion about herd immunity. Herd immunity occurs when R sub S drops below 1. And in order to find out what that means for the population, it's called the herd immunity threshold. And it can be shown to be 1 minus 1 over RS. Sometimes you'll see it written in the literature as 1 minus 1 over R naught because you want to know whether the disease is going to spread. Now, these are powerful concepts. It's a fantastic framework, but it has very serious limitations that, as we're discovering. First of all, I should point out that the caveats I'm going to raise are not in general unique to COVID, but they've been thrust into the limelight because apparently the COVID virus can read, it's read our literature, and it's taken advantage of the limitations. And therefore, those limitations and the necessary extensions of the model are especially crucial for dealing with COVID. For example, the notion of herd immunity thresholds is dangerously oversimplified. This has been the subject of great debate, and there are lots of reasons why it's problematical. Population heterogeneity is one of them. This paper appeared a short time ago by Tom Britton and his group, where they point this out. We think the estimates they have of where herd immunity is achieved are underestimates, but their framework in highlighting this problem, I think, was extremely useful. And our groups, especially the Virginia group and the Princeton group, together with Marty Blazer at Rutgers, have been working on this for a while, and hopefully we'll have a paper soon in which we use network dynamics to explore what herd immunity really means or what it should be replaced by as a concept. So I'm not going to talk about that today. But I mentioned that herd immunity can also vary spatially, and going back to the measles example, there have been outbreaks, and these are the places there were outbreaks in California for reasons I don't understand in terms of vaccine hesitancy around Disneyland, in the state of Washington, in Minnesota, which I don't have highlighted on here, in Texas, because that's where the leading anti-vax person resides, around Chicago, and in communities in New York City. This all happened because of resistance to getting vaccinated against measles, and we get outbreaks there. And so when people ask me, do we have herd immunity against COVID? I said, do we have herd immunity against measles? Averaged over the country, we do, but in various communities, we don't. And that will allow pockets to form, and those can seed outbreaks in other areas. And I'll come back to talk about that when I talk about vaccination strategies and vaccine nationalism later in the talk. So there are a number of limitations, and this is not a complete list, that require extensions of the classical approach. And I'll talk about 4 or 5 of these. First of all, we know with COVID, we're getting lots of new variants on rapid timescales. And that's one of the reasons why we have to keep getting boosters. We've learned how important asymptotic stages can be. We know that immunity can wane, whether it's natural immunity or vaccine immunity, which is why we all are getting booster shots now, or at least those of us above a certain age for sure. Human behavior during epidemics has been largely ignored in previous models, but there are exceptions, I'll point out. But we're realizing because of mask wearing and things of that sort how important that is. I've already mentioned population heterogeneity, and associated with that is the fact that it's not a well-mixed mean field population. There's heterogeneous mixing. There are different attitudes towards things like lockdowns, masks, and vaccination, and they have strangely gotten aligned with political polarization and attitudes towards public goods. So I'll say something about those things. I already mentioned vaccine nationalism, and I hinted at interactions with other diseases, including those attitudes towards vaccination that I mentioned. But it's also the case that last year we had a smaller than usual flu season, because individuals were staying home, individuals were wearing masks. But so, as Sherry Towers and her group have mentioned in their work, when we have low numbers, that may be just building up the susceptible pool. And who knows what that'll mean in terms of outbreaks down the line. So all sorts of complications, all sorts of challenges for the theory. So let me look at them one at a time, and I won't have time to go through all of them. First of all, what about dealing with new variants on rapid timescales? Well, that's also not a new problem for us because it's something we've had to consider when it comes to flu. This is a cartoon of an influenza A variant that can infect us. And there are probably 11 proteins in influenza A, 2 of them on the surface, hemagglutinin and neuraminidase. And you've heard about them, or you've certainly heard about their first letters, H and N. And I mean in the context of flu, I know everybody's heard about H and N. But those are the things that sit on the surface and that interact with your cells. And they take different forms. So you hear about H3, N2. Those are variants on the hemagglutinin and neuraminidase proteins that are sitting on the surface. And the virus keeps changing them in order to escape your immune system. So we continue to get outbreaks. We get the emergence of new mutants due to point mutations, and that is called drift evolution. But on top of that, every 20 years or so, we get a new subtype that emerges. Famously, when the Spanish flu came in 1918, H1N1 appeared. It disappeared around 1957 when H2N2 came in. It was displaced by H3N2. And then in around 1973, H1N1 reappeared, maybe by escape from a laboratory, and has been coexisting with H3N2 ever since, which is why if you get a flu shot, and I hope you do, you will get vaccinated against the prevalent strains, both of H1N1 and of H3N2. Different subtypes arise because of a reassortment event. The influenza virus is made up of 8 pieces that get into your cells. They get into the cells also, for example, of birds, and occasionally they recombine in there. They come out the other end, so that the 8 pieces that come out include some from each of the forms that went in, and that gives you something substantially different from what was circulating before, with essentially no cross-reactivity, no cross-protection, and so the potential for a dramatic escape. It's a puzzle, which we understand a bit, but not totally resolved, as if there's no cross-protection, why it is that when, for example, H2N2 disappears, H3N2 comes in. Is it the decline of H2N2 that allows H3N2 to emerge, or is it the emergence of H3N2 or that suppresses H2N2, or some combination of those? But why should they interact at all if there's no cross-reactivity? Well, probably because there are some generalized T-cell responses early on that do provide some cross-protection, like when you used to get a gamma globulin shot. Or it may just be that individuals who are sick with one stay home and therefore don't get the other. But there's a weak interaction. But in general, except for what happened in 1973, there's no real interaction between them. Now, influenza A, because of these phenomena, has a very unusual phylogenetic tree, as compared, say, to HIV, which you see on the left, which has a number of parallel branches. This is the hemagglutinin chain. In influenza A, you get this curious branching structure, and those are years that you see at the bottom. Each one of those represents a new variant off of it, but those are slight variations, and there's one major trunk. So we've been trying to understand this for a long time, and to understand also how some of the periodicities, appearances of particular subtypes occur. So this is work now 20 years old with Josh Plotkin and Jonathan Dushoff, where we try to develop techniques to predict what the best form of the vaccine would be. We looked at hemagglutinin sequence clusters for influenza A. There were 560 aligned H3 subtype sequences, each one 329 amino acids long. And we did a clustering analysis. And when I say we, I really mean Josh and Jonathan did most of it. And the way you do this clustering analysis is you embed all of the sequences in some space. You define a distance measure on them, where we looked at various distance measures. And then you choose a threshold distance, initially arbitrarily, and you say two things are in the same cluster if they're within that distance apart. And then we do a hierarchical clustering, which is if A in the same cluster and B and C in the same cluster, then A and C are put in the same cluster. And then you vary what the cluster size D is, and you look at the cluster hierarchy, for example, what the size of the largest cluster is. And you add some stochastic element because of uncertainty here, and you get a diagram out that looks like this, where as we change the threshold distance on the bottom, for a while, then look at the top, you'll get no change, but every once in a while, there'll be a big jump. And those are significant places. The first place it occurs, as I'll show you on the virus, is when the virus switches to mutations in different epitopes. And then if the bigger jumps occur when there is a change in the subtype. And so we can go back and trace what the time series are of the appearance of these viral clusters over time. And based on what information was available at the time, we estimate, we had some methods then for estimating where the new mutations would occur and the new jumps would occur, what vaccine should have been used. It's not explicitly topological data analysis, but it's very similar to using topological data analysis, what we do. And others, people have done that, and we interacted for a while with Gunnar Carlson, trying to get some additional insights out of that. So we learned from this that the nature of oscillations in influenza A is on the long time scale, there are those subtypes. That's maybe a 20-year time scale. There are those clusters where we saw those jumps on a 2 to 5 year time scale. And of course, we get new strains every year, which is why we get new flu shots. Well, David Earn and Jonathan Dushoff and I tried to summarize this and look at the dynamics of the cross-protection and ecological interactions between them. And the basic framework from doing that would be to look at a model of this form. Here we only look at SI and R, no symptomatic stage. And the idea is to assume that the population size is held constant, because this is during the particular outbreak or a few outbreaks. There's a death rate delta. It's chosen to match the birth rate in the population. Remember, N is S plus I plus R. But the critical nonlinear terms are the infection rates, beta 1 SI1 and beta 2 SI2, the rates in which two different strains of the virus grow, each with its own recovery rate, gamma. And what one can show from this is there's no stable coexistence. And you can see that because look at equation 2 and equation 3. The only way they could both be in equilibrium, dividing through by I1 and by 2, is if beta 1 S is equal to gamma 1 plus delta, and beta 2 S is equal to gamma 2 plus delta. And that would put two restrictions on S that in general are not going to be possible to match, only if the parameters were chosen exactly to match, and then things would be drifting around anyway. And one can show further that if you let one of these strains reach equilibrium, let's say I1, and try to invade with the other strain, whether type 2 can invade or not will depend upon whether beta 2 over gamma 2 plus delta is greater than beta 1 over gamma 1 plus delta. So one type or the other will win out. It's the type that maximizes beta over gamma plus delta, or looked at it another way, minimizes gamma plus delta over beta, which you can also see is the equilibrium value of S in this equation. So the type that wins out is the type that can coexist with the virus at the lowest level of susceptibles, because then a type that requires more susceptibles can't get going. So it's a very easy way to look at competition between strains. And an early example of this was myxomatosis. European rabbits were introduced into Australia in the early part of the last century. They became pest species. The myxoma virus was introduced, it's a long story, as a control measure Initially, it killed all the rabbits, but quickly evolved to less virulent strains, which means moving to the right here. And that was because there was complete cross protection between the strains, and the strains that killed their hosts too fast did not transmit at high enough rates. So they had lower values of beta over gamma plus mu. And Roy Anderson and Bob May pointed this out and looked at the data in a paper a few decades ago. Influenza A also presents different challenges than some of the other viruses. There's especially different than myxoma because there's incomplete cross protection. If you get one strain of influenza, you can still get other strains. You may have reduced likelihood. You can get co-infected by multiple strains. That's not possible by myxoma. And you get these subtypes that recur periodically on longer timescales. So to look at this, one needs a framework such as this, where instead of just looking, let's say, at S, I, and R, you allow individuals to be infected with type 1 and to recover, and to be infected with type 2 and recover. And then once they recover from that form of the virus, you look at infection by the other type, which can occur at a reduced rate. So we can write a whole bunch of equations. You can see, even for these two strains, it's a pretty complicated problem, a lot of parameters to look at. And with influenza, you've got hundreds of strains. So it's an impossible task. And so there have been lots of efforts, and for example, some by us, led by my student, Sergei Krashinsky, in work with Ulf Diekmann, on the ways to do dimensional reduction of these really high dimensional 10,000 parameter problems. Another complication of the models, the first paper I know was by Dieter Schenzle in dealing with measles, was to introduce different forms of structure, but in particular age structure into these sorts of models. And Carlos Castillo Chavez, Herb Heathcote, and Vigo Andreasen, Wayman Liu, and I, some years ago began to, and we did some work also, just showing you one paper with Ken Cook, who some of you will know or will have known of, to look at models in which we incorporate age structure, mixing, and cross-reactivity to see whether we could, for example, get the periodic recurrences. So for the mathematicians, the equations look like this: age-structured partial differential equations for the aging of the population and translation into susceptible, infected, recover. X, Y, and Z here are susceptible, infected, and recover. I'm not going to take you through this. Happy to send the papers to anyone. We tried to understand endogenous oscillations. We found that the age-structured models alone produced oscillations, but they were damped. So we looked instead at two-strain models. We saw that they could produce oscillations, but they were also damped. But when we put these together, we got two damped oscillators that are interacting, and they cross-excited each other, and you could get sustained periodic oscillations if you added both factors in. But did you need both factors? So working with Juan Lin and Vigo Andreasen, we asked, well, let's look not just at two-strain models, but at three-strain models, and we showed that they could support periodic oscillations. Again, I don't want to go into the details, but if you had three strains with some sufficient measures of cross-protectivity between them, we could get oscillations in the system. COVID also mutates, and so it's quite different than influenza. It's a single-stranded RNA. If you want to learn about the genetics of COVID-19 and the relation to the phenotypes, here's one useful review article I found on it by Musafazada and Gassemi. And it doesn't have those different pieces to get the reassortment event, but it's got its own kind of reassortment. So we have been involved in multiple efforts to deal with new strains. And this work with Osman Yagan and Vincent Poor and other collaborators, which appeared last year. And we've been working with Madhav and his group on more complicated models illustrated here, in which we look not only at the spread of the virus, but the spread of different behaviors like quarantining and things of that sort. And these are multi-scale, multi-layer network approaches that are fascinating. Let me turn now to the evolution of asymptomatic stages. Just to remind you, adding this component back in, asymptomatic infections have been shown to be crucial in dealing with the dynamics of COVID. Here's a paper out of Jeffrey Shaman's group from about a year and a half ago, in which they developed a modeling approach. This is roughly the modeling approach that they developed, in which they allowed for an asymptomatic stage, and they also allowed for age structure in the model. And working with Shadi Sadroy, our graduate student who's been leading a lot of our efforts on COVID, here's the first paper that we published on the dynamic. By the way, Shadi and I had started talking about asymptomatic infections and their importance even before we knew anything about COVID-19. It just seemed like a good strategy for the virus. And there were examples that were known, but then we got the expeditions grant and then COVID came along. I don't think those events were correlated with each other, but we immediately could apply our approach to look at both what the consequences of having an asymptomatic stage and under what conditions would it arise. And so this is the sort of model we looked at. We looked at different strains, just as I mentioned before, I1 and I2, one of which had an asymptomatic stage and one of which didn't. And the time that you spend in the asymptomatic stage was a variable that we could look at from an evolutionary perspective. But just looking at two strains, the SIR model becomes complicated in this way, and you move from susceptible to asymptomatic and from there to symptomatic, or you might bypass that symptomatic stage altogether and then eventually to the recovered stage. So this leads to more complicated dynamics. And then we put these different strains with different values of alpha-1 in competition with each other, alpha-1 and alpha-2. And we use the methods that I described before. Look just down at the bottom, the evolutionarily stable strategies we could compute as those that minimize the susceptible fraction at equilibrium. And we were able to classify the conditions under which we would expect an asymptomatic stage to emerge. Now, of course, this depended on what we hypothesized for the trade-offs between rapid progression, which would allow more rapid spread, and hiding out for a while so the immune system didn't get at it. Again, anybody who's interested, I will happily send you the papers. We looked at a generalized trade-off function, and then we looked at a number of specific forms. In any case, this model shows that asymptomatic infectious stages can indeed evolutionarily emerge, but we knew that from COVID, but we explain why. And also in some conditions, there's bistability in these systems. We extended this, and I'm doing this just so you see what some of the mathematical challenges are, to look at superinfection. What does that mean? That means if you get infected with one strain, it doesn't prevent the other strain from infecting the same individual. And this leads to more complicated evolutionary dynamics. Turns out you can no longer just minimize the number of susceptibles at equilibrium because the secondarily invading type doesn't have to invade just susceptibles. It can invade individuals that are infected with the other types. We looked at this also in a different paper in Interface, where we had two subpopulations in which different strains might be established. What does this all mean for management of COVID-19? Well, first of all, I want to point out that exact timing of, for example, when to get quarantined, when to tell people they can take their mask off, is not possible. This was a paper that appeared now a couple of years ago in the New York Times saying that data showed that delays in lockdown cost at least 36,000 lives, or had it at that time. So led by my former student, Dylan Morris, and Karina Tarnitsa's former student, Fernando Rosina, and together with Josh Plotkin, we published the paper, Communications of Physics, I guess it was earlier this year, in which we computed the following. If you know exactly the dynamics and the number of cases, what would be the optimal thing to do in terms of when to put lockdowns on and when to relax them? And we found the answer to that. But we also showed, and again, it's mainly Dylan and Fernando, who should take the credit for this, that if you were a little bit wrong in what the levels were, and if anybody follows the CDC's COVID tracker, you'll see that there's a lot of obvious variation. I mean, you'll look at it and it'll say one day there were 27,000 new cases, and the next day 110,000. That's just not possible. It's obviously a delay in reporting these things. And so the 7-day average becomes more reliable. But the point is, you can't be exactly sure. And what we showed is, if you were a little bit wrong in applying the optimal strategy, it could lead to disastrous consequences, and you'd be much better off with a second best strategy, which, for example, imposes lockdown for a longer period of time and doesn't try and turn it on and off with the same degree of accuracy. And so in the face of uncertainty, we advocate for a different way of controlling the epidemic. There are a lot of unknowns surrounding immunity that can shape epidemiological dynamics. Another paper led by Shadi and Caroline Wagner on how all these uncertainties, not just about immunity, might play out over the next 5 years. And we looked at various scenarios there. You can't give a definitive answer, much as politicians would like them, as to what's going to happen, but we lay out the range of possibilities. This published about a year ago, I think, in Science. And we looked at things like the strength of immunity, non-pharmaceutical inventions, the effects of seasonality. Again, happy to send it, but you can easily find it in Science. We also showed that why you would get multiple peaks out of it. Another question that's come up, and we have multiple authors on this, as you see again, Shadi is the first author, Caroline the second author, is there was a lot of discussion about if you had limited availability of the vaccine, is it better to make sure everybody gets their, if we're dealing with the Pfizer or Moderna, for example, their 2 doses, or should you spread them around and get a larger number of people getting the first dose? You may remember there was a lot of debate about this, and another paper in Science in which we explore the uncertainties and point out the importance of getting serological data from vaccinated individuals. And finally, in this list, we just published a couple months ago a paper on vaccine nationalism, the idea that let's take care of ourselves first, America first, and worry about sending the vaccine overseas later, if at all. Well, this paper points out the fallacy of that, at least under some conditions, because if you don't protect populations internationally, new variants are going to form that are going to come back to kick us in the pants eventually. And we modeled this with 2 countries. This shows it with 2 countries, a high access region and a low access region, but obviously this generalized. It's crucial for us to be exporting the vaccine so they don't export the new strains back to us. Our approach here was sort of motivated by earlier work that Brian Grenfell, one of the authors, did in dealing with measles, not internationally, but in the UK. But this is a simulation that Brian and Odar Bjornsted and Kappy did many years ago with measles, a video. If you can just look down the bottom, this is just Manchester and London. The point is that if cases keep arising all over the place and you damp them down locally, this is going to seed new infections everywhere and you'll never get control of it. So vaccine nationalism, not a good idea. A couple of other points I want to mention before closing out. Immunity might wane. My student, Luojun Yang, is leading an effort. This is a commentary we published, or they published, I should say, 2 years ago. This was for measles. This was before COVID was a problem. And Alison Galvani and Jeffrey Townsend, who are both part of the expeditions effort, have examined this for SARS-CoV-2, and we're trying to follow up on some of their excellent work today. What about human behavior during epidemics? For example, attitudes towards lockdowns, masks, and vaccination. Well, this is not a new question either. Charles Perrings and his group at Arizona State, with a number of co-authors, including me, including Carlos Castillo-Chavez, and Kinzig, and our former postdoc, Ben Morin, have been looking at what happens if you take into account that individuals during an epidemic, if they're susceptible, for example, will want to avoid interactions with infectious people. I won't take you through this, but I can again send you those papers. Naomi Leonard and I, together with our former student Yongxiao Zhao, and now with student Matthew Chung, are looking at could we use this to develop an optimization approach to changing individual behavior during epidemics. There are a lot of public goods problems in public health. Here's a list of them, and I thank Luojun Yang for this slide, which includes this paper of Allison's on the right about free riding and vaccination decisions. I'm not going to talk at all today about resistance to antibiotics and things of that sort that Ramanan Lakshmanarayan, again, a part of the Expeditions Project, has been leading. Vaccine hesitancy, mask wearing. Somebody sent me this, says, I never use turn signals. It's nobody's business where I'm going. And people are sort of adopting that when it comes to mask wearing and vaccination. The libertarian approach in the country, claiming that mask wearing, whether I wear a mask, whether I get vaccinated, is just about me, we know is not true. If you don't wear a mask, if you don't get vaccinated, you're putting me and my children at risk. Hesitancy, we also learn, is socially mediated. Amazing. Look at the difference between Democrats and Republicans in terms of vaccine hesitancy, but there are a lot of other categories along the way. So how do we change attitudes and how do we change access? Because in some cases, it's not just hesitancy, it's be able to get access to these vaccines. This is a slightly dated slide from YouGov, which points out that there are a group of countries like China, Hong Kong, Thailand, in which anybody of us who have traveled in the Far East over the years knows that masks have always been worn during epidemics, and it's not a surprise that they continue to be worn. There are a group of countries like the Nordic countries in which it's never been a tradition, and it's still not a tradition. But then there are another group of countries like the US in which it was never a tradition, and there's been a transition, a critical transformation, not to the levels of mask wearing that we see in the Far East, but still to a different equilibrium. So we've been trying to model this with Luojun and also with the Virginia group, Madhav and Luojun and I, but the work led by Jiro Q and Baltazar Espinosa, both of whom I think are on this call, on what the dynamics of mask wearing is, what drives it, the role of fear, but also the role of contributing to a public good and protecting others in the society. And that's what Luojun has been leading and works with our group at Princeton, looking at social norms that can either trigger or prevent behavioral change. What will others do and what do they expect me to do? And demonstrating the potential in these models, these are modeling approaches, for a sudden transition in this coordination game from an ineffective equilibrium to an effective one. The models take, just briefly, a multi-layered approach, as I laid out at the beginning, to social norms in which individuals have opinions and actions which relate to the institutions that they're part of or that govern them. Those institutions can also change their rules. And this builds on a framework that Rick Durrett and I started looking at 20 years ago, and that Vitor Vasconcelos and I and others have been using to look at a variety of things, including attitudes towards decarbonization, where one computes payoffs of different behaviors and then looks at the game theoretic approach to these problems. And finally, prosociality is important because it means that I care about you as well as about me. It tends to be more localized, and so we're trying to implement an approach here that Avinash Dixit and Andrew Tillman and I have been developing, in which the population is broken up into the groups. You have a payoff function. You're willing either to keep resources for yourself or to contribute to a public good. You care about other individuals in your own little group, and maybe that's your family group, but now those populations are connected up. So this is work we've just started, and we'll say more about it. So I'm going to conclude. I only have one or two more slides. I wanted to highlight the mathematical challenges that all of these questions raise. There are issues in dynamical systems and stochastic processes, in graph and network theory, in the development of agent-based models, in which the Virginia group has been in the lead, in optimization and control, and in game theory, as well as possibly in topological data analysis. So going back to where I started, I still would argue that Kermack and McKendrick, their equations provide a firm foundation, but I hope I've convinced you that we really need an expansion in multiple directions to deal with the challenges we face today. And there are a lot of exciting opportunities. Almost any branch of mathematics can be useful there. So thank you all very much, and I'll be happy to take any questions.
Excellent talk, Simon. I'm going to let others ask questions because I get a chance to ask you other questions oftentimes, but I let Erin coordinate this part of the discussion. And we should have Simon, if you're around for a while, we can continue the discussion. We exhaust the questions.
I'm fine.
Okay. So,
all right, so we'll have Moshe Vardi first and then Jeff Townsend.
Thank you, Simon, for a fantastic talk. But what strikes me is that when I'm kind of part of this, we hear more about the 1918 flu epidemic. It's become clear when you read about 1918 that the issue of pandemic management, the kind of the social and sociological, even political aspect of this, already came up in 1918. But somehow when you look at the research, it was all focused on epidemiology. And only now we are realizing that actually looking at the management aspect of it and the sociological aspect, even the political aspect, is also incredibly important, you have shown. But that work seems to be incredibly recent. And I'm just struck by the fact that for a century, we just have ignored these aspects of dealing with the pandemic.
Yeah, I mean, that's true. I mean, that's the point I was trying to make here. Certainly people were aware of some aspects of the management back in 1918, but I was very young in 1918. I don't remember too much about what was going on then. The political polarization, I just have never, I mean, maybe it was there. I haven't read about it before, but you're absolutely right. The modeling until recently, and I mentioned work, for example, coming out of Arizona State, and I guess Balthazar, you were also part of that group looking at it. That's a fairly recent development, looking at individuals' behaviors at all and how they might be coupled into it. So you're absolutely right. It's a mystery as to why they didn't gain the attention that they did before, but maybe it's because things have just suddenly become so politically polarized. I think it is. I mean, it is the case, certainly going back to smallpox vaccine, which had a lot of risks associated with it, obviously, that was very controversial. And there were people who refused to get vaccinated there. But as far as I can tell, it never really made its way into the modeling until recently.
And the number that I found most striking was the level of resistance among healthcare workers in your different groups. This is, to me, just absolutely astonishing. I mean, only thing I can do is bang my head against the wall.
Yeah, well, what's even more astonishing to me than the fact that some healthcare workers refuse to get vaccinated is the fact that in some cases the courts or politicians don't understand the degree of risk that confers on their patients. When I used to lecture just on things like influenza or HIV, I would raise the question of, I mean, this is one of the earliest network questions I could think of other than the spread question when it came to disease. If you have limited vaccine, whom do you vaccinate first? Do you vaccinate, and this question persists, do you vaccinate first the people who are at the greatest risk? Vaccinate first the people with the greatest capacity to spread it, the nodes in the network. And that would be healthcare workers and sex workers. I mean, those are the people, depending on the disease, who are most likely to spread it. And if you're interested in controlling the disease, those are the people that you've got to get to first. The idea that a healthcare worker, when we're dealing with influenza, would ever refuse to get vaccinated just didn't occur to me. And if that were a rare occurrence, okay, you would let those people keep their jobs, but you'd keep them away from patients and let them start looking at the accounts or something. But when you have large numbers of people doing that, you can't do that. So it's just incredible. You're absolutely right.
Hey, Simon, I really enjoyed your review of all of those topics. One of the things that sort of struck me a lot as I thought about this disease as we monitored it over time, and I think this is not really a new insight, but it just brought my attention pretty strongly to the point, is that the evolution of this disease in terms of its coming and going, all the phases it's been through, it just looks nothing like the outcome of an SIR model of disease, right? You pair that up on one side, it's a single increase and then a decrease, and we've seen just waves and waves of this. And I don't think that that's actually that unusual. And so one of my questions for you is just, one of the things I've been sort of trying to aim myself at a little bit more is maybe abstracting myself a little bit from, or maybe I'm not sure if abstracting is the right word, but focusing a little bit of the kind of modeling that I've been doing to try to look at just, what is the effect of this on some short time scale, given there's a certain level of spread, as opposed to trying to model the whole epidemic. I'm just wondering if you think that's a way forward that we can think about mathematics of disease spread a little bit more, less of like trying to map out the whole trajectory and more trying to figure out what the factors are, and just maybe it's almost a physics approach of just saying here and what can we do to change it.
Yeah, well, I think you're absolutely right. My students know that when they're starting to work on a problem, I tell them my approach was modeled because I was a Baltimore Colts fan years ago, and they had a tackle named Gene Big Daddy Lipscomb, who was all pro. And when he was asked what the secret to his success was, he said, when the ball is snapped, I rush into the backfield, I tackle everybody, and then I sort them out and I keep the guy with the ball. And that's often my approach to these problems, which is, let's get a list of all the things, all the complications. I've given you an impossible list today. Even dealing with influenza and the multiple strains I pointed out is an impossible problem. But all the things we've mentioned today, vaccine waning and human behavior, those are all potentially important, but I think you're absolutely right. What we have to do is now sort them out and find out who has the ball. We have to, in a particular situation, when we're trying to manage on a particular scale, we've got to find out what are the crucial factors that are driving the dynamics on that time scale. So I don't have an answer to that, but I think you're absolutely right that that's what we have to do. And unfortunately, we don't have Gene Lipscomb around anymore to advise us on doing this.
Hi, that was fantastic, Simon. Especially, I hadn't seen all of your most recent stuff, and I wondered if I had read your stuff with Karina Nieborg on social tipping points and social norms, and we've been working a bit on governance for disease management. So, if one area wants to manage the disease one way and another area wants to manage it a different way, there can be this big tension because there are spillovers. And I wondered if you have ever thought about what the social tipping points would do in a sort of multi-jurisdiction type problem.
Yeah, so that's great. That's wonderful. So what Kat's talking about is that we've done a lot of work in different contexts, actually, it had nothing to do with disease, about the importance of social norms and driving behaviors and the potential for social norms to suddenly change. The most dramatic example was foot binding in China, but we've seen examples in terms of attitudes towards smoking in public places here, hopefully gender equality and racial equality, although less of that than we thought. The idea, first of all, that you might have competing norms within the same population, or the interesting notion that Katrina's raised, that you have different jurisdictions in which there are different norms or laws that are under, and how do they interact with each other is a great question. We have, in terms of the former, we actually have, I've just edited together with Helen Milner and Bob Axelrod and Charles Perrings, a special issue of the Proceedings of the National Academy it'll be out in about a month, on the dynamics of political polarization, which has some relevance to this. But the point you raise is an obviously important thing to look at. I mentioned some of the work that Shadi led on looking at the different dosing regimes, and, for example, in different jurisdictions. But I've obviously thought about the interactions of different social norms in different places and the leakage between them, but we haven't done anything on it. I think it's a great thing to do, and I'll be really eager to see what you've written on that. I will mention one example is I remember when I was an assistant professor at Cornell 50 years ago, one of the other more senior professors, a Greek immigrant talking about what he called about being a marginal man, that he existed in two cultures, but most of us do. One of them when he would go home to be with his family, and the other when he come to the university. And that's not like two cities, but even within one jurisdiction, one may belong to multiple groups. We've done some work, by the way, which if you remind me, I'll send you about what happens when individuals belong to different groups. It's a little different than what you're saying, but the cross memberships means every time I make a decision as to what I'm going to do, I have to take into account all the groups of which I'm a member. You know, my parents might have not approved of this, but in the university, that's what I have to do. So how do I resolve that? We should talk about that because that's related. That's a little different, I think, than what you're saying, but it has some relation to it.
I've been listening to this and I'm just really, I don't know where to begin. So let me just say this, is if you think of agency as nothing more or less, not very naively, think of it as mediated behavior. And you think of it as having modularity. So there's a sensing piece, an interpretive piece, an action formation piece, like a range of actions, and then actually an action. All the traditional problems of constraint of behavior by the external world, the importance of semantics and context and stuff become really just inherent to this notion. And so you wind up with sort of a zero intelligence kind of argument. If you think this through, you say, well, okay, how do all these mindless bits and pieces come together to mediate behavior? You wind up with basically a discussion of something along the lines of, we have systems that regulate systems that don't regulate themselves, and systems that, don't regulate systems that don't regulate themselves. And you can wind up with a pretty simple argument, which I won't go through right here, but you wind up with the obvious paradox of saying, if a system that regulates systems that don't regulate themselves, does it regulate itself? And you wind up with this motivation to type theory of agents. And so you're saying there must be types. So you come up basically with what Simon is saying. You wind up with systems that are type distinct, that interact on an agent level basis according to type, and they're within agents of other types. And this seems sort of obvious, but it also seems sort of lost in the discussion. It has real implications, both to theory and implementations. There are metaphors of this or sort of echoes of this in combat modeling. In World War I, basically you could use Lanchester equations and so on, and you could calculate exactly the rate of fire and how many artillery pieces you needed and invent machine guns and do things and just draw land combat to a complete draw until everybody just they couldn't win, they just quit. And what happened was warfare morphed over the next 100 years into involving every single person on the planet. So now we wage war against every single person on the planet in every form of warfare it is, whether it's nuclear weapons or terrorism or what it is. And so you have this complex collection of agencies, whether you want to call them social or engineered or whatever they are, and they're interacting. And I just think that the connections to really some old things in logic, the embodiment of logic in modern computer science, these kinds of agent-based network models that we have now, and these aggregate models, whether they're Lanchester equations or epidemic models, the kind that we see, all of these things are in our face now that say local mediated behavior. First of all, it's type, so you can call it social or individual. It matters. It's actually part of the system. There's no way around it. And all of these things where we try to avoid it because it's somehow not science or something is behind us, and we have to deal with it. And I think it's incredibly, it's scary, but it's also incredibly reassuring that it's being just so directly raised as an issue in this kind of work. It's absolutely essential in this work, in economics, and lots of other things. It seems to me we're confronted with it. But the political issues you raise and modern warfare itself, I think, are also major indicators of how important this topic is. I can't imagine going forward with this discipline without having a much more nuanced and much more serious theoretical approach to thinking about agency than we currently have.
Just to add a point to what you said, because your comments stimulated this thinking, is the problem of systemic risk and contagion with the growing interconnectedness of the world for a variety of reasons, not just people traveling more, but because we're so interconnected on the internet, and because, as you talked about, the weapons of mass destruction, their range is increasing. This breaks down the features that make our systems robust and resilient. Bob May and George Sugihara and I wrote a paper in 2008, even before the financial crisis, saying the increasing interconnectedness of the financial system was setting it up for a collapse. And so I think with the growing interconnectedness of the world, if we don't catch up with making governance more international, we're heading for a lot of COVID-19. And I don't mean just in the infectious disease world.
I think, Simon, I should maybe ask Moshe to say a little bit about his pet topic right now, which connects back to the story on resilience, because it's something that he's been very strongly advocating. And I think it connects to the story really nicely.
Oh,
Moshe, do you have to say a
absolutely.
little bit? Yeah.
As soon as I saw this, I said, exactly, it's another manifestation that the efficient policy is not resilient.
Right.
Because it will not survive very small perturbations. And it's okay to be less efficient, but because you are facing so much unknown, we need to learn how to build resilient model, how to just change the way we do analysis of such things. Because very often our approach is built on optimization, efficiency.
Yeah, I mean, that's the paper I mentioned, led by Dylan Morris and Fernando Racino, points out that the efficient solution is dangerous there, and that that trade-off comes up over again. By the way, it relates to a fundamental question in evolutionary biology, which is essentially the exploration-exploitation trade-off, or the question of how much do you invest in addressing today's problems as opposed to maintaining the capacity to adapt and deal with the. So yeah, Moshe, you and I were supposed to have a series of discussions of this. We better get back to them, because I agree with you. This is the fundamental trade-off that we face in addressing sustainability broadly.
I want to acknowledge Kate and Mike and Cecile's work, and others also, but on starting this scenario modeling hub. And I bring it up because it's very deeply related to talking about the short-term forecast that was a question that Jeff had asked. And I don't know whether Mike or Kat want to say a little bit about it, but I think it's very relevant. It's a different style of approaching this problem than the typical forecast. And I think it might be a good thing for them to say a little bit for this group, if you folks are willing to.
Sure, I can mention it briefly. What we do is we were, with COVID just starting out, the problem was there's immense uncertainty. And so using ideas from Mike Runge's research and ideas about multiple models, we pulled together the idea of running a scenario hub, using multiple models as if they were individual experts, and we could solicit their opinions about what would happen, but as a modeling team rather than as an individual. And it's a way of dealing with the uncertainty that might hinder either good scenario projections, or in my case, I would be very tied to making that about decisions. And so we've been running a scenario hub. Madhav's team contributes, one of 9 or 10 that contributes, and we're in the 10th round of this for the CDC. We've just contributed to the ACIP decisions about 5 to 11-year-old vaccinations. And basically it's a way of dealing with the uncertainties that are particularly egregious at the start of an outbreak. And Mike, I'm sure you want to add to that.
No, not much to add. It's Moshe's comments about efficiency is not the same as robustness. And part of the stuff, Simon, that you were talking about today is about capturing uncertainty broadly and not being overconfident in our understanding of the dynamics. And so, we've been really trying pretty hard in the scenario modeling hub to, through the multiple models, really capture as much uncertainty as we could and fold that into the sort of decision-making enterprise.
And we did have one example in an early, our case study actually had 17 independent models or distinct models that contributed. We did a sort of pilot study for it, and we had a hospitalization objective. So most of it, we were talking about minimization of cases or deaths, but actually we did a risk threshold analysis on hospitalization. So there might have been strategies that would have very similar expected hospital capacity, but very different risks of exceeding hospital capacity. And that would be really important to a manager that wants to know, what's the chance I need to send my patients elsewhere or set up a field hospital? And so you're not worried about what the average sort of median or outcome is. You're actually worried about the risk of exceeding some sort of intolerance threshold, which would then reflect this argument about robustness versus optimization.
So one thing I was thinking, Simon, is maybe we'll invite you to one of the meetings that they organize, and perhaps we could decide on one of the scenarios in the future once you both agree on the writing Simon has had. I think we are starting to go to scenarios that are 1 year long, which is we never did before for a while. But I think some of the questions Simon raised today are now in that regime. Talking about endemicity of the disease, or talking about how these things live through from an evolutionary standpoint. And I think perhaps that's the kind of questions we can undertake in upcoming scenarios. So if Simon, you're open, we'll invite you in. Kate and Mike should.
Yeah, great. I'd love to join.
Well, and the round we're running right now, Simon, is on waning immunity crossed with boosters, for example. So
great. Any other questions? This has been absolutely superb talk, and the questions. I think the questions are equally important. Simon, I'll just close from my side by, first of all, again, thanking you. It fits our biocomplexity view. It's an amazing talk. You have an amazing view because it spans a long period of time, and I've not seen too many other works that has connected ecology in the broadest sense, not just humans and plants, but all the way to economics and social systems and back to diseases. The arc is absolutely wide, and so it fits the concept of biocomplexity the way I think we would like to embody it here. But just amazing talk, and hopefully we'll have a chance to hear you again in maybe a little bit time. We have 5 years going, so we'll do this again. But thank you everyone here for attending. Absolutely nice talk. I think a lot of people would like to hear on this. So on that note, unless there are any other questions, I would like to thank Simon once again and close the meeting. Thank you.
Thank you all.
Thank you very much
Absolutely.
again. Have a good afternoon. Take care.
Bye.
All right.